still having issue with Zoom sometime, but that last week I was presenting things through Teams and I felt so confused.
It is, it's tough to use
It's
Teams.
so different. All right. I don't see anyone walking in, so we can walk through this quickly.
Everyone must be planning to watch the recording, I guess.
Yeah, but it's probably fine. We are on the we're on the O, right? So I don't have to unmute myself, I guess.
Yeah, the owl's going, you're good.
All right. Okay. Um, so this is meant to be watched for the recording as well, because it's just like, revisit of some of the things that we've been talking on. So if anyone is late or anyone, have to do some other stuff like during this call, it's fine. And I'll be,
Big class today.
yeah,
Yeah, come join.
yeah. So I'll be talking for just like 10 to 15 minutes, answer the quick questions, and we'll just move to the real discussion time. So what I'm going to talk today is about the Golden Gate assembly, which is one of the molecular cloning method. And you don't need to know. Connected tomorrow? Yes. So this is something that we plan for the lab at MIT for the CRISPR stuff, where it's like, easier than Gibson assembly even. But because like we haven't touched that yet, so I just want to bring everyone up to the same speed on what it is. So, again, I try to like revisit it on this slide every time we talk about anything that like most of the time we engineer any biological system, we're going to engineer at the DNA level. And on this DNA, there are multiple ways to make the plasmid or the plasmid DNA. And the three key assembly that people have been using in synthetic biology, that might be using the digestion ligation, where you just cut thing and paste thing all over the place. And that was like the old school style where like, in 1975, that was like how the first DNA cloning has been done. Gibson is one of the tools that we use a lot for the Scalars assembly. Most of the time when you do Gibson assembly, you need to design a primer. And because like the oligonucleotide synthesis has been made a lot cheaper and faster these days, that's why people do a lot of these Gibson assembly. However, the third one, which is like a Golden Gate assembly, where you take the parts from different DNA pieces and stitch them together in the organized way. That was like one of the like golden method on the molecular cloning in SynBio, because you can technically have a lot of the building blocks made, like pre-made, and then you assemble them to make a lot of parts. So just to walk through a little bit of these, these are figures that I got from Agene. And you see that, the way that the restriction ligation work is by, if you have two pieces of DNA, most of the time it's going to be like a plasmid with insert and a plasmid backbone, where you cut parts of the backbone using these, DNA cut sites, and then you cut something similar from the donor plasmid. Then you take the insert, most of the time smaller, but sometimes bigger. If you have like a big insert, then it's sometimes even bigger than the backbone. And make sure that the sticky end is matching each other. So like similar to like how you're playing Tetris, it's gonna be it was showing up all the time on whatever cloning you're doing. And you have the new plasmid where this red piece, which is usually called multiple cloning site, was replaced by the desired gene, like the blue thing right here. This is just the same thing, just another schematic that I got from all the sites. I've shared this slide, and you can take a look at whichever figure you like. They're explaining the same thing. So these restriction enzymes and ligation system is pretty convenient, and also relatively cheap compared to the other parts. But there are some limits because you need these restriction sites. So it's not a routine anymore. The routine one is the Gibson assembly style, which the MIT students here has done on one of the lab class here. And I think like of you might have heard about the Gibson assembly for many times already. It was um, spearheaded by Daniel Gibson from Check Institute, which is a colleague of John Glass. I think John also stopped by to chat with us in the class in the genome synthesis part. So how it works is that like you have the overhang where it was the same as the other part, and there will be like shielding process to make it similar to the sticky end that was generated, and then just do something similar to the restriction ligation. But these parts were generated from these double-stranded DNA. This is just another style. Well, maybe it's not here. It's fine. I think Gibson, you know it pretty well. So the last one is a Golden Gate, where it was relying on one of the spatial enzyme called type 2S restriction enzymes, where it recognized one piece of the base and cut adjacent piece. How does that make it different? It just means that like those cut sites, the sticky ends can be something else apart from the recognition site, so you can use that to program what it should be. So let me go ahead and I'll show it to you, and you can get and ask a question when I show all the slides. So these type 2S, you can cut them and you can make them, like let's say sticky end A, and the other part will be sticky end B. And if you make sure that the sticky end B and B are compatible with each other, then you can sort of like assemble them. This is like a zoom in. When you use BSA1, the recognition site is this GGTCTC, and they will not cut right away after the GGTCTC. They'll skip one base and cut whatever 4 nucleotides right there, and that will turn into the sticky end. If this part of the DNA is TATA, then you have that whatever complementary to TATA sticky end. If you have another piece that was cut by this one as well and has a TATA sticky end, then it will find each other and do the ligation. So most of the time, the Golden Gate will be a fusion of the scar that was generated by the BSA1 or some other restriction enzyme cut sites that was type 2S, where it cut outside of their recognition sites. And if you put the cut sites into the piece that you're going to remove it, you sort of like don't have any scar of this Golden Gate, type 2S restriction enzyme anymore. So what you see right here is that, if you cut these insert fragment A, and you have this orange sticky end and the red sticky end, and you cut the insert B to have these red sticky end and blue sticky end, these two red can find each other and do the ligation, similar to how the restriction ligation work. And the orange will find the orange part on the backbone, and the blue will find the blue part on the backbone. So this is like, how it can work further on, where you can program these, orange, red, or blue part. I'll go back to your question.
I don't really understand the difference between this and like other restriction enzymes.
Yeah, so the restriction enzyme, you use the restriction enzyme that was recognizing most of the time 6 base, and you cut in the middle of those 6 base. That means whenever you do the ligation, you will always have the restriction recognition site at the time. The difference for the Golden Gate is that you put the recognition site on the right-hand side, and you cut, and you don't really need the right-hand side DNA piece anymore. Then the final product will not have the cut sites. And by that, it means that you can sort of cycle it, where like if you get the final product, it will not be cut anymore. So you can do something similar to the PCR by keep doing digestion and ligation. If there's a product, it will not get cut anymore. If there's anything that was like go back and self-ligate with themselves, then they will be cut again because they still have that recognition site. So that means this Golden Gate, you can sort of like cycle them. And first time, maybe you have like 50% of your backbone turned into the product. The second round, maybe another 50% of the remaining one, and you get closer to the 100% over and over. So that's the reason why it was very powerful, but it needs a lot of pre-design.
So the enzyme, it's like it's a specific enzyme, right? So it has a specific cutting site, or
it has a specific recognition site, but not cut site. As you can see right here.
Cut wherever you wanted that.
You can design, you can specify what the cut site sticky end would be based on the position of the recognition site. It's not that you can make it anything, right? Because it has to be 5 base away from the recognition site, and it has to be on the right-hand side, because this is like not symmetric anymore. Most of the hexameric, like the 6 base recognition enzyme, were based on something that was palindromic. So, either reading from the top or from the bottom, they're the same. This one is not. So it's either going to cut to the right-hand side or the left-hand side. Yeah. So by that, you can sort of like, make these programmable sticky end. You can make it clear when you're designing things that, this is going to be the promoter part, where the left sticky end will be, AGAG, and the right-hand side will be ACAT all the time, so that you can keep changing the promoter and the terminator. And you can sort of like, if you have like, let's say 90 plasmid to begin with, you can assemble, 100,000 variants from that kind of assembly. So this is like useful in many ways because you can do it in the library as well by changing, different parts. Let's say you have like all 6 parts remain the same and you change the promoter part. That means you can do the promoter library screening without having to do any DNA synthesis, as long as the starting Moclo kit has that. They call it Moclo by like, molecular cloning. Modular cloning is another word because it's modular. They can be interchangeable between each other. Right. So these two references are like, one of the good references that you look into. Like the 2015 ACSML paper is on the yeast molecular cloning yeast Moclo YTK yeast toolkit. Is one of the most famous Moclo paper. And the other one is the recent Besa subtilis, where you have to design in a way that like you need the E. coli part, you need the B subtilis part, something like that. So, if you take a look, it will tell you some interesting thing about this Moclo. So, what does it mean? Why do we care about this CRISPR? So I'm going to go back a little bit that like, we are trying to use CRISPR to do gene programming. So I introduced about the CRISPR activation in the last recitation. So if you have multiple kinase and control different genes, then you can control the gene expression of different genes at the same time. In this case, I sort of show that we have multiple kinase, right? So in this case, if you want a combination of like strong, then you can just try to build it as is. If you try to build like weak, you can also do that at the same time. But imagine that there are like 64 combinations. You have to make, all of them. How are you going to do it? Are you going to do like 4 and then add another 4 and add another 4? That's what we did when we start. But after we finish everything, and because like we are trying to grow further and further, it's getting like very difficult to program it, because like if you have to build, let's say, you add another guy in it that control 4 levels, then 64 times 4 is going to be 200 something. And you don't really want to prepare all those 200 pieces. So it's a lot easier that you specify what you want and then assemble what you want as is. So in this case, I'm gonna I ordered the fragments that representing these 4 levels of 3 set of guidelines so that you can do in the lab by assembling through the Golden Gate assembly. And what we have is that we have this backbone that has GFP. So you see that red part is the recognition site. We'll cut it, and when the recognition site, those LS and LE sort of like recognize each other and assemble into the final one, then there will be no cut site anymore, and it will remain as your final thing. If it's a 2 insert, then you have to like program it so that like the linking part are compatible. You can go up to many parts as you want, and the efficiency may drops a little bit as you keep increasing the parts, but it can, like, theoretically it can go up to 50 parts for the assembly. So specifically we're gonna have 3 guyanase of and those 3 guyanase, they are the same set, but they're gonna have 4 different levels. So what I have handy right now is that I'm gonna mix all 4 and all 4. So they're gonna be like 4 by 4 by 4 combinations in that transformation that we're trying to do. But in the lab tomorrow, you can also plan to do that yourself by like pick if you want the strongest, the weakest, or whatever as one of the 3. And that's going to be the final one where you have like 3 guyanase controlling whether it's going to be high, medium, or off target, strong, if you want to express only one percent protein out of 3, something like that. All right. So you're going to be able to arbitrarily make one of these circuit based on the combination that you want, or if you want a couple, you can also plan to add a couple variations. You can keep one of the first gyne constant and keep the last one constant, and just like add 3 variants of the middle one. That means you can have 3 possible outcomes So technically, in that case, if you have the proper reporter system, you can actually figure out the colony that you got in the library, which one may have what kind of output. So, that's it. And this is how I want to, show another molecular assembly tools to combine with the toolkit that we have for the tuning. You can choose whether you want to go high, medium, low. If you're, don't really want to try too many things, I think I order a couple of the single guide insert where it's going to be very efficient. And those are the CRISPR interference, like to repress to repress this gene expression, and you can try to use those for the repression of some of the fluorescent protein that we have, like the green, red, and orange fluorescent protein that we have in the lab. So you can try that as well. All right, so that's the intro for the plan tomorrow, and hopefully that could be fun. It's gonna be quick, but it's gonna be a lot of waiting time.
Yeah.
Right.
Are these slides something that you've already shared or can share easily?
I can share easily.
Okay.
I'll put it up for you right away. All right. Any question in the chat or any question in the room?
For the node side, I know some nodes might be interested in trying to run this lab in like maybe a week or two or something.
Yeah.
Would they potentially be able to order, like, would they order the same DNA that you have listed on the ordering sheet, basically?
Yeah, they can do that guide RNA, but they will need the plasmid from us.
Okay,
as
so
a starting point. So it's not going to be that easy.
Maybe better
Yeah.
not to do that.
Because like the plasmid that I have ready is a plasmid that have the dCas9 and the actuator and the recipient, like GFP dropout. We call it GFP dropout because we're changing GFP to something else.
Mhm.
So because we have that plasmid ready, we can just do it here right away. But if any other nodes want to do it, I'm not quite sure if like our deposition, like maybe my PhD lab deposition on that gene is going well or not. But if it is really okay, then it can be shared, or we can figure it out. How do we share this backbone easily? Because there are two things that we need, right? There's a reporter plasmid and the backbone that you can use to change the guide program to something else.
Yeah, I like Eric. That's a good suggestion. Maybe we should make this a cloud lab unit so it's easier enough and get this out to nodes.
So I have designed, a couple carotenoid biosynthesis parts as a reporter, but I didn't design the CRISPR stuff because it's kind of huge and it's going to take time for the synthesis to undergo anyway. And because I have one something that I clone and I have already, so I don't feel like spending maybe 2K to synthesize like the last gene.
Yeah, fair enough. Do you happen to have like a lab protocol or anything we're following tomorrow, or just going off of your knowledge?
Yeah, I do have that drafted, but I think I may have not linked into the system yet.
We'll figure it out later.
Let's do it.
I just was checking it
during the time tomorrow, and everyone can take a look on what we have, and we'll add the pictures that we got tomorrow into our lab, because this is a new lab because we combine the bioproduction and CRISPR together. So we have the CRISPR and bioproduction nodes of the lab, but this one is going to be a little bit different. Technically, it's going to be simpler, but it will require both background knowledge on both things.
You had me at simpler. Yeah, that sounds great. So we'll update you all on the actual like lab protocol soon, like later this week.
Sounds good. All right, I think that's it for that, and I will check the chat.
Suvin, I don't want to put you on the spot. I know you're not feeling like optimal.
Oh no, let's do it. What I want to do is I want to go over informally like a lot of the requirements for the final project. So that's coming up pretty soon for both the in-person students as well as the committed listeners. So I think it would be good to spend, let's say, 10 to 15 minutes looking at some examples. Again, what you're expected to do, and additionally, like maybe take like breakout rooms for a couple folks on Zoom and in person, just so you guys can go discuss briefly about your final projects. Does that sound good?
Sounds great.
Great. Yeah, and apologies I couldn't be there in person, a little bit under the weather, but yeah, let's get started. So I've already sent some of these links, so you can again find everything on the individual final project webpage. Hopefully everyone can see this. But again, if you go to individual final projects, as you all should know, you should be able to find your documentation and what you're expected to do for the remainder of the course. So again, you guys have been all working on different and pretty cool final projects, but really what we want to do is have you guys focus on 5 key elements of these. And a lot of these are centered around the project aims or section 2. So especially for those in person that are aiming to complete parts of the project, the goal that you're expected to have is again three main aims, right? You're going to have an experimental aim, a development aim, and a visionary aim.
Well, that's where the
The
terms came from.
Yep.
When I added it in this document. Oh, I see.
Yeah, we actually changed the structure like quite recently. Like this was a new format that we introduced last year, and we found pretty good success with it. And we'll show you some examples. But really, the main goal is for you to think very extensively about aim one, about how you actually would go complete this. So this is akin to like maybe like a first or second year grad student, like for example, completing these assignments. And for ICE, I know a lot of this might be seem trivial, but it's like some of these are really to get you again into the shoes of how this kind of science would be conducted in the real world, and especially in a lot of these lab settings. And so again, as you guys have already done in practice, we really want to make sure you have a mastery or at least some intuition of the core aims and how you'd complete parts of it. For example, if you're working with, let's say, some certain DNA orders, how would you go and measure these, characterize, and so forth? So really, you want to spend the time to go and thoroughly analyze and explain these. This can end up taking quite a few sentences. So we expect to be around like 3 to 4 paragraphs for these. But a lot of these are like you describe them and then you spend time writing a lot of the background. So a lot of these also involve quite a bit of background as we were talking about. And so one of the things that we'd like to see, especially in this year, is how you're able to look at some of this literature. Now, for whatever major problems you may be solving, like whether it be like, for example, solving allergens, detections, biosensors, and a plethora of other topics, we know that there's a lot of literature that has gone into supporting this and getting an understanding of the field itself. So what we are expecting from students is a thorough analysis of like what you've looked at in the background, whether it be, for example, research papers, an innovative approach to your project, and a lot of the impact that we have. Again, we want to make sure that we can emphasize the roles as a scientist being able to explore the field. And this is something that you want to do that, for example, frankly, like ChatGPT can also help you with this, but we really want to see a lot of those papers that you've gone through and like analyze. So again, everything I say right here with the big context is that if I just click Control C and put it in ChatGPT, I know that it'll give me answers, and I know that everyone here in this chat does know that too. So, but the main thing we're looking for is really like a more thorough analysis of, okay, you're able to go retrieve these. What do these help you do? Like, for example, maybe it's that you found a specific sensor that you have that is studied in literature or that needs some improvement in some factor. That could be something you go and focus on and say, oh, this could be what we use to base off our examples. And I know very well in advance that I'm talking a little bit vague and like higher level, but we'll go and show you some examples as we walk through the papers. So this kind of background is really critical for as you write through your analysis. And I think this is something that where while ChatGPT will eventually improve on a lot of these, it's like something you can still do. And we do expect you to go through and look at some examples, like for example, maybe some of the figures you have as well as other descriptions. As you
Yeah, I
write
just say
this,
like
yep, go for
wherever
it.
possible, like please include like images and like pictures. Like it just helps so much during the final project presentations. And like even if it means like you didn't actually do anything in the lab quite yet, like if you could generate images that of like what the lab work would look like or what the results would look like, that's super, super helpful. So just to put that
Right.
out there.
Yep. And a lot of this can also be looked at, for example, like the applications or your output. Like what exactly are you measuring? And so what have people measured in the past as what you can use for equipment? Any questions there about the background and names? If not, great. Then I'll keep going. We can feel free to interrupt, folks. Like you guys can just speak up and unmute and so forth. So the other aspect that's also quite critical is the ethical implications. So I know we haven't spoken a lot about this since probably like the first few classes, and this is something where you want to spend some time to also go through and see, let's say like your system is implemented, what potentially could go off target, or what could be an issue that you foresee, and how would you mitigate it? So you can think back to a lot of lecture one topics where we had a couple speakers discuss the ethical implications of, for example, let's say releasing a specific like biomolecule or some sort of a coli that is genetically modified. How would you protect it against unwarranted effects? So these are something we especially ask you look into in high detail, and we have a lot of really good examples from the homework people have done about week one to help you with this. This is something where again having like those Venn diagrams and examples from like for example like how let me see if I can pull up an example. Um, for example, these kinds of mitigation strategies really come in handy. So charts like, for example, like this, and I'll link this in the chat, where you can see here that folks like you guys would have had to go and analyze this and assess based on different conditions of like, let's say a final project and certain options of whether this enhances biosecurity lab safety, and so forth. So you can use this to your full advantage. And I highly encourage you guys to go back to these and use parts of this. Again, these weekly assignments were intended to help you construct a lot of your final projects. So don't be shy of like going back to prior weekly assignments and pulling these. Moving on to a couple of the key experimental technologies is that once you have your aims and once you've written them down, you can again not only use Claude to refine these, but we want to see a very detailed experimental plan. So this doesn't just have to be like paragraph dumps, but rather you can explain like a certain vision for it and then explain how you'd implement it and show a lot of the figures. Like one of the best ways that you can go and explain your experimental design is go on BioRender or go on these other tools that can help you generate some of these flow diagrams and use that. Even a simple flow diagram of showing boxes and arrows from PowerPoint is infinitely better than having a stream of text. And again, all of what I'm saying will show some examples, but this is something where as a scientist, you want to have other people be able to understand, frankly, the design pathway and the choices you made because of this in a couple minutes or so after reading it. And what we want to do is like as you're doing this, we want you to relate back to the lab. So when we've gone through a lot of our different experiments we've done, for example, the very final one we're doing with CRISPR tomorrow, you'll notice a lot of the different aspects of the labs involve very similar methods. For example, pipetting is obviously one of the major common ones, but also plasmid preparation. You might be working with cell-free and like several other protein design elements. We want to see that. So we want to make sure that you include a lot of these and showcase exactly what you want and what you want to use, as well as how you were able to like utilize some of the experimental plans to use it. Again, you don't have to repeat all of the technology from there, but it would be good to like showcase some of that in here. And lastly, we ask you to expand really on the methods you use. So, for example, like how you want to use some of these companies to help you analyze these results, and as Ronan said, expected results in section 5. So maybe some example graphs, some output that you measure. For example, is it like a fluorescence reading? And even some plausible data. Like let's say you're trying to compare two different outputs. Like you might say, oh, if you are building a biosensor and it detects a certain type of like molecule, what would you expect the output of like maybe like some like GFP be? Like you could show a graph that says, hey, this is what we expect to be in these conditions versus in a baseline or a negative control. So we want you to, again, just remind you to think similar to those terms of how you convey these as a scientist. So that's a quick summary of the document. Again, you've probably seen this a million times, but I wanted to just pause there for a minute to see if anyone has questions here. Again, this is very critical to make sure you follow this, not only for grades for MIT and in person, but also for the committed listeners as well that are completing a final project.
I'll also just mention like this is a pretty like structured write-up format, but like how to grow is about being creative and like don't feel limited by this structure. Like, have fun with this, add your own sections and like just be creative, be your own person. This is just a rough outline. Try to hit these like bullet points though, like for sure, but then, also go off on your own and show your own work. So we're super excited for your projects. Ooh.
Cool. Then if no other questions here, let's show a couple example ones. So I sent a couple in the chat, but we'll show you one from last year as well as one from the year below, or the couple years before that. And then finally, I'll wrap up with how the committed listeners will eventually present. So first up, we got Smello World, which is done by Cyrus Clark, who you guys might know very well from who hosted Hard Hacks at MIT. And his project was focusing on DNA storage as well as how to look into, I guess, the smell itself. So the way he went through the project itself was discussing a lot of the background behind data storage itself, setting up the context, and looking at some of the innovations here. And so what he did really well, though, was a lot of the experimental analysis was very laid out. For example, how he wanted to like showcase what he wanted to do. For this case, he wanted to make a toe hold switch and use a specific trigger to report an outcome. So diagrams like this are perfect for immediately conveying like stuff that you do and don't have to be that complicated, for example. And the goal of this project really was to design a specific coding sequence that can be like recognized by a toe hold switch, then output a certain fluorescence. For example, GFP, or in this case, a banana scent, which is kind of his whole goal of like, for example, Smell a World to use some of the senses. And one of the things that he was able to do was discuss a lot of the DNA design as well as the method to do so with some of the methods that we've used from the class. For example, how he was able to go and create his different plasmid with the toe hold switch. So you can see that he has a couple diagrams of the toe hold itself, the general mechanism of how it works, but then also some analysis of like the different steps he has. Additionally, he's also included a lot of the work that he has to generate some of these results through in silico work. So some of these outputs are here. And for example, the benchling designs that he used for some of these to hold switches. Now, at this point, you guys should have already made a lot, like especially for the in-person students, like you guys should have already made a series of different benchling designs for your final project. So these are perfect to include in something like this, where you already have examples of what your output would be. For example, your plasma design, as well as how you've actually went and designed them. So they're very valuable images to already put into the report. And eventually, what this led to was a protocol built that you can go and actually analyze the specific plasmids. And the rest of the write-up focuses a lot on the designs of how he made those toehold switches. Again, another novel aspect that he did that we want to see from students where your design input and a lot of the work you've done to create these constructs will be quite useful. Let's see if we can jump to one of the final ones. Here. So in the measurement aspect, like one of the examples we're looking for and really emphasize is example results that you'd have for a project like this. And in this case, Cyrus went ahead and explained, for example, using some previous literature, what these kind of expected results would be for a to-hold switch. For example, you have a switch here, a positive control, as well as the actual trigger for it, and what this mechanism would look like. For example, if you have this to-hold switch, you get some output in whatever you're measuring, for example, GFP. This is the kind of thing that we really want to look for your final projects, where you're able to clearly emphasize that this is the expected output. And you'll also have 7 minutes to talk about this too. So like you can walk through in your final presentations and projects a series of this different measurement, explain the rationale behind it. I know in this case, I'm like going through very quickly with this, but in the actual talk, you'll have more time to go through and analyze these, and then also talk about the future work. So this is one example of it. There are other examples that we've done in previous years where, while this wasn't based off of the aims, you have other projects, for example, this, which is about bacterial shading and using different mechanisms of coral to actually create different outputs based on light itself, the reaction. So I'll link it here as well. But you can see here that this is also another head TA previously where Laura made a lot of really cool diagrams, explain a lot of the structure regarding this genetic switch, and also additionally included her benchling designs for the changes she wanted to make with her plasmids, as well as a lot of the output with specific genes of interest. For example, she borrowed a lot of the ones that she used from one of her Gibson labs, a male sepia gene, which you might recognize, and looked into how can she use it to modify her genetic circuit. And additionally included some expected results, for example, a ladder here that would expect after some sort of DNA purification or an assembly, as well as certain aims that she's included. But I encourage you guys to go through and also read a lot of these examples because there are really some wonderful visuals people have made. For example, how our system would work, as well as the actual mechanism in 3D that you can take advantage of to really convey your thoughts. And again, a picture is 1,000 words. So please look at these and look at how these like pictures are used to showcase some of these final projects. Because again, this is frankly really cool to see a lot of these come to life. And while you might not be able to get to that stage, having these images will definitely help you there. So I'm going to pause there for now for any questions, so forth. But again, really cool projects that I cannot do justice in describing, and we've linked a few of them at the very bottom of our discussion for past final projects. But again, we have 7 years of different projects that you can go over, and hundreds, if not thousands at this point, of designs from committed listeners and final projects.
Yeah, that's true. I'm really excited to see what you all come up with, like website wise. So that'll be kind of fun to see. Although I think
Yeah, I
actually,
think
Stephen, are the committed listeners and everyone global presenting on the slide deck?
yeah.
They're not actually presenting their web pages, right?
No, but that's also something where we can also chat about a little bit too. But it's still you for the committed listeners, like you still will need to complete your final project web page though, to my understanding. Um, yes, we should definitely look at all 7 years. How to get there, doing that in the next 10 minutes is gonna be impossible, but
Yeah,
um
good luck tracking down anything past like a couple of years ago. It's hard to find the
yeah.
links.
Yeah. Like again, like you just go to How to Grow's website and like pull up a bunch of different projects. Like even from like when I took this in 2022, you can see there's a lot of like really cool projects people have done. And I will give another example too. But
A lot of the Notion links have expired, so I think it's
that's
really
the
good we have this
problem. Yeah.
better infrastructure now. So if you do put effort into your website, it'll actually be like
This is
persistent
interesting.
across time now. So
Oh,
that's
this is so
nice.
unfortunate. I
Yeah.
wonder if we can find a way back because I know we definitely have them, a few of these, but figure out like which ones let work. But there's a lot of really cool ones that also look at protein design. But again, we have tons of different material that are just here scattered across. So definitely take a look at them. And I know 2019 still should work because a lot of these were based off of like GitLab. So we have a lot of them here, like for example, like Pat's, where you can go through and see some of the final project work. And again, there are troves of different design work that people have, lots of cool figures of people like experimenting with some of these, changing, for example, like the Chrome Force structure. But again, some of these bio render figures that people have where you can see the whole pathway. And the last thing I'll touch about briefly is the marathon of the committed listener sprint. So this becomes a giant marathon at the end of it. So it is a multi-hour presentation for committed listeners, and I've included this link as kind of an example of how chaotic this can get. Like for each node, you'll have a presentation of around 3 slides and a timed 3-minute presentation, and it goes by fast. But you can see here that my computer is already struggling to load all 261 page slides of this. So there's an insane amount of figures of from really cool people that have worked on this last year. And for folks that are presenting the community listeners side, I encourage you to take a look at this because a lot of these are really structured about presenting your aims as usual, some of your experimental analysis, and potentially expected results for some of these. And there are hundreds of different projects here that go through these with great detail about, for example, targeting specific peptides, or again, some of their benching design and evaluation. So definitely take a look at some of these and see if we can use these as inspiration when you go about doing these. Again, all of these links are here linked above, and the world is yours for being able to try to look at some of these because I can guarantee that for almost every one of your projects, someone has already explored a very similar one that you can look at as an example for how to structure it. And I think I'll stop there.
Yeah, if you end up doing lab work, like, and you feel comfortable sharing like pictures of what you did, or like even you in the lab, like just include that. Like, it's all so much fun, you know? Like, we want to see the community aspects that we're building and how it
Yeah.
grows.
And even if you can't, that's totally fine. Like I would not stress out too much about it. Like your grade is not negatively impacted by that. Like we know things happen and like with our ordering side, so don't feel too stressed out about it. But
Oh yeah,
yeah,
even on the MIT side, like there are certain reagents or things we're not going to get in time, so it's just part of the journey.
yeah. I think Ronan, I'll hand it back to you. If you want to add anything else or ask for questions. Do we want to do the breakout room right now if you want? Like I think that would be good for folks to like
You
do
know,
that
I
in the
guess
last.
I think we do have enough people. We should do breakout rooms.
Yeah.
Yeah, just so people
So
can
let's do
chat.
that. Yeah. Let's take like the next like 5-10 minutes and then break out into groups of can you make it into like groups of 5 or so maybe and like have people discuss I'll answer this question right after, but maybe in like a minute we can do that with like 5 people or so per group. Does that sound fair?
Oh,
Or do we want to
you
do
think we
larger
should use
groups?
kind of smaller five
I feel
people
like that's
group?
the case because it might be harder for people to discuss. So, and for continued future research, are you talking, I guess the question is depending on like whether you want to continue the research at your node or is it like for like final project presentation wise, like what to explain there regarding the question in the chat?
Yeah, I was just mainly wondering if there's like any kind of support to continue our final projects and like what are the things that all past students have done. Kind of
Got
just
it.
wanted to get an idea of that. Thank you.
Yeah. So I think actually a few of us in the room, like I think for Broden and Alex, like you guys have both continued on with stuff for how to grow in the summer. So it's like it's definitely very possible and like very much like encouraged. It really depends on the node. Like I think for us for MIT, it's like it's pretty decently flexible. Like we still have some reagents that we can order and buy throughout the summer and beyond, but that's something you'd have to have like the node to node conversation, like with your node leader, whether they have the resources to support it the at least for MIT, I know that we've had typically around like 3 to 5 people continue with how to grow over the summer for various things, either through final project work or using the space for other things. But again, probably a node-to-node conversation, which is also where your final project result, like your explanation there, kind of helps because if you set it up quite well for what you want to do, for example, let's say your reagents didn't arrive in time, or for example, you have some really bigger aim you want to do, but need some sort of like startup or like some sort of incubator to help support it, there might be an option to do so.
Yeah, some of it will be node based. If you're interested in doing like cloud lab research stuff, I will definitely be like fully into that like during in the summer months. So like I really want to focus on like the 20 things to do with that cloud lab storyline and showcase like a bunch of projects. So I will definitely want support and collaboration on that. So we'll figure out like the details, but like I'll probably put out like some, I don't know, like sign up or something at some point so we can all get on the same page about that. But we'll probably do like weekly meetings and stuff like that. So there'll definitely be a lot of ongoing research into the summer for sure. Okay,
That's
I'll
great.
do the breakout
Thank
rooms
you.
so we can all
Yeah.
chat about projects. Good question. Thank you for asking that. All right, let's open these rooms for a minute. So yeah, have a second to chat about final projects, and then we'll bring everyone back in a minute.